So, the point is, there are three speakers. They're all good friends. And I find very difficult to introduce friends. No. So to make it easier, you can read. Ramesh has been, I think we know each other for more than 30 years. That was in JNU. And from that time only, he was different. He created a special center from UGC for genomics facility. And that went on in different forms even now. And those days, running a sequencing facility and things like that was remarkable in 90s. So I know him from that side. And I also remember making good papers. For example, the pyruvate kinase, JBC, and those kind of things. So he's in wonderful and very illustrious career. So I say you don't give a scientific talk, but give something relevant to today's theme. He very kindly agreed. Very difficult to catch him, but he managed, and he's promised to stay till tea time. Doesn't eat anything, so even if he stays, he will be useless. So, Ramesh, please.
Screen. Does whatever you speak. So in the meantime, they fix it. Very good afternoon to all of you. So nice of Professor Prasad inviting me over here. And his colleagues, the organizers of this. Thank you very much. Although I had the opportunity to see MIT campus, Gurgaon campus, both Noida as well as this campus earlier, but this is different. And Professor Prasad calls you here and invites you, takes care of you. Thank you very much.
So before you start, this I forgot to tell. Ramesh was the first person who came to visit MIT, especially whole university, as a UGC visiting team. And he was very tough. And during the subsequent years, he has been very, very helpful in helping us bringing some programs right here. For example, this gateway teaching, he was one of the experts. I don't know how much he helped, but I think he's helped a lot. Thank you, Ramesh.
I was the chair of the committee.
See.
Well, I'm going to speak on shaping biotechnology, education, and research in India. And I'm just going to give what opinion I hold. You heard in the morning the Secretary DBT, and I was wondering, sitting out there, that, well, you will see the same theme running through my slides when I talk about it. But the expression is going to be slightly different because I'm going to be a little critical and futuristic and try to understand or convey to my student friends over here that what exactly is required and what has gone wrong and where that till now we have not been able to achieve what we had the potential of. We have achieved a lot. There's no doubt about it. So let me have the first slide, which says, well, on the white, it may not work. So it says we take pride in what we have achieved. I mean, definitely, we take pride in what we have achieved, but could we achieve something more? So it turns out there's a report which BIREC, you're familiar with this, it's a DBT arm, which has a report, Bioeconomy Report 2024. I took data from there, but there's a report which is 2025. So I prefer to keep this report to tell you that from that report onwards, in a year's time, we have made substantial progress. So that's a happy note. The substantial progress has been made in the area of bioindustry, biopharma, bioagriculture, and I have clubbed this bioinformatics, bioservices as bio-IT and research services. So in all these areas, we have substantially gone reasonably high in terms of the national GDP when we compare. It's more than 4.2% now. And what I have put here as ecosystem of startups, I have put 800 over here. It has gone beyond 1,000. Comes the question that if we take pride in this, and the question is, could we have done better? Then has education really served this purpose? I mean, what you see there on sites, and we have done very well, if you come to think of it, we do not have much data available with us where we can say, well, academia and industry put together, how much have they contributed independently, and how much have they contributed together by collaborating with each other? My hunch is not much. If this has happened on industry scale independently, and some academic input here or there, very random. So it's not systematic, it's not sequential, it's not well organized, it's a knee-jerk, it's ad hoc kind of risk. But it has happened. Fortunately for us, we can boast about it, so it has happened. What has gone wrong? If I say it has not happened between academia and industry to that extent, where things have gone wrong, I have drawn this pyramid over there. Biotechnology, to my understanding, as I know of it in different university systems and the different research institutes who carry biotechnology program, it's more of cellular molecular biology. I used to say it in a very lighter vein, long back, maybe 20, 30 years back, and joke around with my friends that all institutions in our country are becoming CCMB. So everybody does cellular molecular biology. Now, I'm not non-serious about it, but at the same time, I want to convey a message through this to my young friends over there. Biotechnology is not only cellular molecular biology. Biotechnology is whatever knowledge you have gained in cellular molecular biology, you have to bring in technology to convert this into some tangible outcome, some product, some application value. Now, in all biotechnology setups, our education is geared in a manner that is conventional. So we have to somehow move away from this conventional, not leaving aside the fundamentals which are essential. Because if you do biotechnology, in fact, when it was introduced at 10th, 12th level, I somehow was uncomfortable because biotechnology should not be started at that level. That is the stage when you should be studying good physics, chemistry, conceptual level physics and chemistry, mathematics, and biology. So that when you come to the graduation level and post-graduation level, you apply that knowledge to the biology and generate new knowledge. Why new knowledge has not been generated in our country if we have hundreds of institutions and we have now more than 1,000 universities? New knowledge generation is somewhere there's a paucity of that. I'm not saying it's not there. There are pockets, but there is paucity. The scale we are in the country of institutions, in fact, I should say it here and qualify my sentence by saying the human resource we have had in the past, the expertise we have had in the past is par excellence and comparable with any country abroad, whether US or Europe. Unfortunate part is we have not been able to thread it together, tie it together, provide that kind of a leadership role to see to it or have clarity at that level. What do we really want? What is our priority? Where should we place those priorities vis-a-vis our country or country's priority? Or societal, because everything has to be seen in societal terms as well, besides doing good science. So both ways we have suffered. So I have drawn this pyramid for my young friends over here to understand if we want to do well in education and biotechnology, let's keep this in mind. Our fundamentals have to be very clear. Conceptual framework has to be there for everything. It has to be interdisciplinary, because without that, you will not be able to bring out that application value to the knowledge which you are studying. And then, of course, the purpose of biotechnology is that you should be, as a student, you should be equipped to translate that. So their technology part has to come into picture and translate it to a product or of application value. So the question here is, in this pyramid, is the workforce which we are creating in the country, which is competitive globally and locally important, because now this is the phrase being used that you have to do local as well as global. So it turns out that in biotechnology, somewhere it's very patchy and not our learning outcomes. Although UGC introduced it many, many years back, I used to be on that committee as well, for learning outcomes, how they should be defined. And learning outcomes are challenging still, and we need to have that art of converting, translating knowledge to a product. That is lacking. Here, teachers also need to be trained. So unfortunately, this is another caveat which I would like to add, that we have been from conventional areas and we have been following that, we have been passing out in that. That's our comfort zone. Whether it's research or it's education or biotechnology in our country, we have lived in our comfort zones, and it has been very difficult for people to come out of those comfort zones to marry biology with technology and make it biotechnology. I'll just give an example as a case study. Like in the morning, you had some case study, and it was related to medical biotechnology or health and disease, which the Secretary DBT presented. In this, I won't go into the details because you all are familiar with the kind of information which is coming out of precision medicine, the word which was used. It has become buzzword in the past decade and a half or two that we use in medical biotechnology, one of the areas, sub areas, and sub area of health and disease. Have we been prepared systematically for handling precision medicine in our country? I have drawn a list on the left-hand panel over there. Try to go through that, and I'll just pick up one or two topics from there. If a student of first is conceptual understanding, clarity. Are we clear about when we talk of health and disease, of epidemiology? Unfortunately, the word was not used in the morning, should have been used. Today, it is molecular epidemiology. Do we understand molecular epidemiology well? Well, you will not be able to do precision medicine. You have to be a good genome person or omics person, whether it's transcriptomics, metabolomics, or proteomics, or whatever omics is there. You may be good at reading it, but in terms of application and trying to understand it at the population level and try to understand it in the whole country, you need to be familiar with epidemiology. And epidemiology, unfortunately, has been very poor in our country. We realized it in COVID days, but then we picked up very quickly because there was a lot of pressure, both politically, socially. So it worked out. But epidemiology has to be understood. And today it is molecular epidemiology. I mean, you need to understand that if you are different from me and a given population group gets disease, preferentially a different kind of a disease, and another population a different disease, that I need to understand this from molecular epidemiology point of view. The whole etiology behind it. No, that is simple. Etiology is simple, but then I have to take it to the point of care also. I have to understand it from diagnostic to, I have to have preparedness for intervention also there. So are we ready for that? So I'll come back to that intervention part of it, and that what is going on world over in this direction. I didn't want to go through the whole list. In fact, when you talk of epidemiology and you get a particular SNP or a variant, genomic variant, which is of importance or significance in a given population group where there is a preferentially susceptibility to a disease, then you have to understand it in evolutionary terms also. That when peopling has happened in the whole globe, when they scattered here and there, how did it emerge? And the treatment is designed that way. Now, try to understand, there is a lot to it. I could lecture on this probably whole day, but it suffices to say what all is एक खिचड़ी भी पकानी है तो उसमें ingredients कौन से अच्छे होंगे जो ये tasty होगी. So forget about the khichdi. It's not a khichdi. It's a systematic knowledge base which is there, and you don't want to make a khichdi out of it, even if it is tasty. So from evolutionary perspective, from epidemiological perspective, from molecular epidemiological perspective, in order to even do biotechnology is important. That means there has to be a conceptual understanding. I have not gone to quantum physics and application of quantum physics over here. I have not gone to AI and how predictable it will be, or using machine learning tools over here and how they will help to predict this, and then probably wet lab will establish it, then I'll use it in community. So I have not gone in that particular direction. Today, the situation is that this morning you heard about the technologies which are there of gene editing. The gene editing which is being attempted even in our country at certain centers is ex vivo. There is gene editing going on everywhere abroad in most of the places is in vivo. Even the delivery systems using nanoparticles, how they have delivered it in vivo, targeted it, and then corrected the defect. And CRISPR-Cas is familiar to you all here in the audience. There is base editor, prime editor, twin prime editor, and they have advantages, base editor as well as prime editor, in terms of not introducing, wrong or not either inserting or deleting wrongly in the DNA sequence here or there. So kind of off-read sites. The success story is this, through base editing. Again, a CRISPR technology which has been used. Now this is live in front of you. Where do we stand? Why haven't we achieved this, which is happening here? For a urea cycle disorder, it has been corrected and the baby is doing well. And this is in vivo. And there were 11, I put over there somewhere, there were 11 teams coming together. Now, what does it bring out? For my young friends over here, it brings out that technology alone, knowledge alone, which we have in the country, it's there. The 11 teams coming together is an issue. So how 11 teams got together and worked collectively for a cause and a correction through base editing, the same CRISPR technology, but in a different form. Because even in CRISPR-Cas9, you have Cas9, which you have to modulate because of a variety of reasons, and people have done a lot of research in that area. But it's beyond that. It does not introduce double-stranded breaks in the DNA. So it takes care of that. So through this, I have put down over there the concern now in our educational system of biotechnology is, if I have to talk of medical biotechnology and health and disease, is that what takes us to do the same in our country, India? Manpower, know-how, scientific understanding, technology, we have all at hand, yet it doesn't work. So talking about something and projecting it in a particular manner as if the country is matured to do it, there are gaps and we need to identify, and I'll come towards the end, or how those gaps can emerge and be identified at your level and how they can be corrected also. There are success stories. There are success stories in agriculture, in agricultural sciences. There are success stories in animal world, in veterinary science, biotechnology. There are success stories of that kind, but not to the scale as is desired in our country. The scale is very different. We have to see to it that how do we attain that particular scale. This was talked in the morning. In research terms, this is what we propose. So we are an year old, and I will not talk about it too much because it has already been introduced by the secretary in the morning of DBT. I will only talk of that it's talked about in terms of bioeconomy. In my first slide, I showed you all that has been done in either biopharma, bioagri, bioindustry, or IT related biotechnology, all has generated a lot of money. It is bioeconomy. Now that we have framed it in a particular manner by terming it very, attractively, it's wonderful. But then biotechnology is not bioeconomy alone. And let's not at least I hold that opinion, that we should not be only be drawn by bioeconomy aspect of biotechnology. It's an offshoot, it's a product, byproduct. So let's take it as byproduct and not kill the science behind it, not kill the innovation behind it, not kill the new knowledge generation behind it. So we have to balance that, which doesn't get balanced by BioE3. I wish Secretary DBT was here. I would have conveyed it to him. Otherwise, I will not be able to convey. Maybe my friends who meet him, they will convey him that Ramesh had this opinion, which was countering yours. And probably Modi ji's also, if it reaches him, that there has to be, otherwise what will happen in 10 more years to come, a decade to come, our bioeconomy will match in $1 trillion with global, because global is being projected at $30 trillion. And we'll also reach what is our projection is $1.3 trillion to 2 point something odd trillion dollars. It will definitely reach, because the biofuel by itself is generating so much of economy, which you saw in one of the slides of Secretary DBT. The problem is that's not what we want alone. So my student friends over here, you have to take a leadership role in the future. Be aware of a balance and don't be drawn by catchy things, catchy presentations, or catchy statements, or catchy slogans. Have some fundamental sense about what biotechnology should do for our country. We have to generate new knowledge. That's very important because we do not have discoveries in our country. We do not have inventions in our country. We have some innovations in our country which get converted, and our industry friends are able to convert that into some technology. In fact, I remember some of the, okay, let me come to that. I'll point it out over there. A small shopkeeper also does a business. And I have put that at the end over there. A small shopkeeper is very useful, does it for survival, everyday economy. So there is economy of kind being generated by that particular. But is that what you want? I mean, it's fine for that particular setup. I mean, it should be respected. Respect has a different connotation. What we are talking about is at a scale of the country, what we should be doing in education and research in biotechnology in future. So I'm pointing out those gaps. So we have to think of both local needs as well as global. And there has to be a SWOT analysis in terms of strengths, weaknesses, opportunities, threats. This should be done regularly in our educational system to evaluate. And I will end for this particular slide by saying, which I didn't want to, but I was pondering over that, whether I should talk about that. The areas which are 6 areas which are pointed over there, and the areas which are pointed out here also in this, these 6 areas figure, at least 2 or 3 areas figure in 2011 report of Department of Biotechnology. So 2011, 2025, 14 years. My question is, what have we done in these 14 years? Can we do stock taking, the same SWOT analysis? Can we try to understand in terms of that, the outputs of those, the money gone in that, evaluation of that? So accountability factor has to be brought in trying to understand that. That's how we will progress. You youngsters have to remember about that. And we all have to be accountable for that. There are, this is just a casual slide, platforms. There would be various kinds of platforms depending on needs, vaccine platforms. And one of my friends sitting out here probably would appreciate when during COVID-19, we did not have a platform in the mandated institution in our country. And the platform, vaccine generating platform, should have been there because it has been made for that particular purpose. It's a mandate of that institution, but it was not there. So it's fortunate for us that the companies, Bharat Biotech and others, they came into picture. Others did manufacturing job, but Bharat Biotech was the one did from, you may not say great technology, but did some job. So we need to have platforms of different kinds. They have to be thought over. That is biotechnology. That has to, students have to be trained on those platforms. They have to understand, hands-on exposure has to be for such purposes. Then only you will be able to move forward and translate. Otherwise, how can you imagine how to translate it? It's all bookish. And that too, cellular molecular biology. It's not biotechnology. Now, this may be a busy slide, but then I took a lot of time to understand and put it there. So you have education and research, which we have talked about. You have knowledge base getting generated from there. It's also clear to us. It should be conceptual, but also advanced, because we can't be staying at one place. We have to move on globally, locally. And there has to be skill sets, which I have put certain questions for. Do we have those skill sets? What we have skill sets for, we have maybe we are semi-skilled, or I won't say unskilled, or we are not completely upskilled. We need to get upskilled for that. So this has been done in the morning also. Biotech incubators in higher educational institutions was talked about. You have one over here, I heard in the morning. But what kind of that setup is there? Because I'm familiar with these biotech incubators, and wherever they have been given, the DST has given a lot of money in different institutions for biotech incubators. What kind of projects are they supporting? What kind of technology is emerging out of it? To what extent it has gone up to industry? It's like patents. You know, you file patents, you have a list of patents, and you put it in your biodata, but hardly one of them has not even seen the light of the day by getting converted into a product. So we have to question. You youngsters have to question, know about it first. Then second, not only question and be negative, no, be positive, but identify what should be done or as an alternative. So for these semi-skilled, reskilled people, you have jobs, careers over there as technical people, mid-level people, but you have to go to the higher level also. So how can you find that niche for yourself if you choose a career of that kind? And there has to be measurable learning and translational outcomes, which I have already talked about. There has to be product of relevance, high-end research, simultaneously generating new knowledge, I have talked about. But there's a challenge, which I have put towards one end. The challenge is availability, accessibility, affordability of quality also of modern biotech-based diagnosis, prognosis, interventions. Because I picked up as one of the case studies, example, medical biotechnology, health and disease. So I thought I would only pick up, otherwise you have to talk about agricultural, industrial, and whatnot, microbial. So it's a plethora of things. So focusing on this and conveying the message is, do we have for last man in our country to get in an affordable terms, accessibility of those? Intervention, forget about. Intervention, I showed you the baby who has been corrected for urea cycle disease and is doing well. There is another adult who has been corrected for some other problem, that is hypercholesterolemia. So there is another, so there have been, for genetic diseases, there have been corrections done through CRISPR technology. It's in vivo. These corrections are already gone. We will be probably 20, 30 years hence, we'll think about it and then do it. We'll still do ex vivo, that way in cancer situation, not genetic diseases. So nobody is doing in genetic disease. Cancer, yes, ex vivo, not in vivo. So there is challenges which you youngsters have to realize, that what do we do for making it available? I'll just pick up small examples over here to make you understand. Suppose if I'm doing mass spec and I see some unique peak coming out of it. Now, there's no time left. Otherwise, I would have posed this question to you. How will you make it very simple so that at a primary health center level, which is called wellness centers, again renamed. So those wellness centers, you're able to provide for a villager who comes and accesses a test. because you know this is a unique peak which I have seen in a high-end lab, mass level, mass spectrometry level, and I have now this. It can diagnose. I have established it. You can convert it in a very simple form, an assay, and detect, at least in that population who comes over there as patients, and then detect. Similarly, in sequencing, can you evolve something very simple which can be done in the same fashion, and you can detect mutations? Yes, you can still do it simple ways, and you can detect that particular change. So you have to think in those terms, that even if you are not generating a new knowledge, at least you be innovative to some extent in years' time, that for all 800 diseases, 1,000 diseases, you have these systems in place for a primary health center or a wellness center to provide. So there has to be someone, two people out of you youngsters, one knowledge generator, another industry person, a startup who does this. Only available knowledge is being packed in a manner and executed so that a poor gets benefited with very little money to spend. So that is one challenge. And there is another one which interests me all through, and I have been talking about for decades whenever I have been on certain committees, is a comprehensive understanding of any proposal or project which you have to execute in the country. And that is that you have to generate matrices. Matrices is that you have a matrix of how many villages and districts and states and UTs, union territories, you have in the country. Then you have to understand how many institutions, higher education institutions, schools, colleges are there in the country. You have to understand how many students are there, researchers are there, area of expertise, very well defined, nuanced. And then national priorities have to be put on that. You generate these matrices, one about the other. Once you have a group of matrices, you apply machine learning tools and you try to understand where are gaps, which are relating to each other. You can also use AI tools, because after machine learning, you have AI. So you can have, and then you can set conditions that what is my national priority? I want to do tuberculosis, say, for example. What already information exists over there in whole of our country, where it has not reached, where it has reached. Every information is, if I have 8 lakh villages, 6 lakhs in villages, and urban villages is 2 lakhs odd. So I have for everything a data. I'll find a lot of gaps here and there. It will mean a step ahead towards applying biotechnology. It's only a first step. You realize that by generating a matrix of this kind, you realize ultimately I have done nothing. Nothing is there because there are too many gaps coming over there. And then you have your futuristic projections are defined by these generating these matrices, putting upon each other and trying to understand gap areas, where gaps are there which need to be attended, and technology need to be developed. It will also be posing research questions. You can do it on all levels for even understanding what we have to do in the country. Now, what has to be done? I'm probably towards the last slide. Some things have to change in the policy and architecture. This is one. And second, at the base, I have written over there, change also has to happen in the character and mindset within the higher educational institutions, its academic and administrative ecosystem. Now, these two have to go hand in hand. We may be very hushy about things and everything is goody-goody. But when it comes to brass tacks, it's taxpayers' money we are spending. And we have to, you youngsters have to realize that it's used usefully. It reaches whom it should reach. I may afford in a private hospital getting admitted and getting treated, but you have to think of a person who can't even think of that, who doesn't have this at his disposal or her disposal. So we have to change. We have to think of changing policies and architecture of that policies. And then within institutions, in education and research, we have to see that the mindset changes, and both academic and administrative structures. And I have put on the side, we have national priority. Our national priorities is never ending. I have known in funding agencies, all the funding agencies of a country, that there are priorities which are set 40 years ago, they still remain the priority. What happened in 40 years? Why this priority was not taken care of? So I have put over there, never ending. See, these are national priorities, which are never-ending national priorities. And group of people decide these priorities. they make it very important. I call them dominant voices. Say they become dominant voices, and these dominant voices determine what should happen. And what is happening is that we are not able to filter through the strengths in our country. I have just put it there, pooling of strengths. I think there has to be a system where pooling of strength, whether inimical, not inimical, no, I don't like your face, you don't like my face, it doesn't matter. For countries, in the interest of the country, all strengths have to be pooled, and we have to see to it that how we take it forward. Appropriate expertise. केले की जगह संतरा रख दिया, संतरे की अमरूद रख दिया, अमरूद की जगह कोई रख दिया, zoology की कमेटी में botanist रख दिया, botanist की कमेटी में zoologist रख दिया. So appropriate one, and then within those people who take decisions, they have to be knowledgeable. They are taking decisions which matter for our future generations to come. So if they are not knowledgeable, they should simply say, I'm not knowledgeable about this. I can't take a decision, but they take a decision. So national priority, appropriate expertise, pooling of strength, accountability, I have put a question mark. No accountability till date for anything. Facilitation till delivery. You are supported, but you're not supported to the hilt. Did you deliver? It has to be seen that if there's a potential, it has to be supported till they deliver. Mission mode, again, we have a lot of missions. There have been programs through our funding agencies, fortunately. I'm not saying it negatively, they have done very well within the resources they have. Country has progressed, there's no doubt about it. My only concern is we could have done much better within the same resource, which was available to us. So what was mission mode in the past is forgotten. What was mission mode in 2011 appears in 2025 as if a mission mode. That hurts because 14 years, what happened? And then there has to be outcome-based evaluation for everything. And this has to happen at institution level also. Institution also has to be very open-minded. They don't have to put it on papers, glossy papers, to show the public, to show the universe or the globe that we are doing very well. At ground level, the strength has to be there. It will automatically show. You don't have to show and do your PR to show others. It shows up. It will show automatically. Thank you all. Lesson for my youngsters. I told youngsters. Okay, go ahead.
Wonderful lecture. You really presented the true picture that we have today, away from all the fads and other things that we talk about. My problem is that how do you rectify these in the true sense?
I have given solutions also simultaneously.
No, but then can these solutions not be implemented? If not, why have they not been? You mentioned that priorities were defined 20 years ago. Still nothing has happened. So some way accountability has to be fixed. What is your suggestion on that? How do we go about doing it?
The first solution is, if you have spoken and I have spoken, these two voices and two more voices here, one in the middle over there with strong voice, if these 5 voices put together say, it matters. It definitely matters, because it doesn't matter. And since it has been posed, see, we in India, I'm sorry to put it this way very candidly to my young friends over here, everybody is not above himself or herself, whether they are in administration or they are supporting people through funding agencies. If they rise above their own self-interest, things will get rectified. That is one of the solutions. Second solution is that, because if I have personal axe to grind, I'll simply keep quiet. I just keep quiet because मेरी fund ही नहीं मिलेगा, मुझे project नहीं देंगे. See, I have been marginalized whole my life, yet I did well. I could have done much better. But anyway, that is when I have to tell my personal story, or I will write some story. So people can be marginalized, but you can't annihilate them. So they will speak. But there are others who have always served their interests. they have to speak because they are in those groups or where their voice matters. And they can influence the dominant voices because dominant voices can't be the right voice. The problem is, I have said it in bigger, I had to give some convocation address and I said, same thing. I said, dominant voice cannot be the right voice. So I think let's get together. I have no problem. Let there be a draft of just 1 page. Let there be half a page, but it has to be evidence-based. See, it cannot be a feeling because if it is a feeling, then it's problematic. Then it means I don't like a person, I don't want that person, it's like that. But if it's evidence-based, as I have tried to do in my slides, I could have done it much more because of the time constraints. If it is evidence-based, like I gave the example of 2011 and 2025, it's grossly there. Same area, same language. Picture is different in 2011 annual report and 2020. But my young friends, it's not a disheartening message. It's a message that where we have triumphed, we have done very well as a country, we are united. But at the same time, we should learn to question things which have gone wrong. We should also bring in the accountability factor so that I also follow the accountability factor and I ask another person also to follow that accountability. So the voices have to be put together. Yeah. I'm sorry, I'm taking time. So
Yeah, so this is a very provocative topic.
I know it is. But
we can go on.
Yeah, but this is important that's why I'm trying to give also final message to my young friends. Don't take things negatively. Please be always positive because if you're positive, you will spread a positivity around you. If you're negative, then you will be cynical. I'm not cynical. If I was cynical, then I wouldn't have done research as I was able to do whatever way. So don't be cynical, but be straightforward, have a spine. And when it's appropriate, then only speak, okay? When it's not appropriate, don't speak because it will not be valued.
Thank you very much. No, wait. Okay. So next speaker, another friend, very difficult to describe. Dr. Deshpande, please come forward. A great scientist, spent his whole life in NCL, Pune. And we have a connection, academic connection. For some reason, I was his DSC examiner when he got the degree. So since then, we are together. Good friend. And after NCL career, he has set up not a startup now, full company, Green Biotech in Pune. And they are giving a lot of agri products, pesticide management, etc. and in market. So he's a very silent, revolutionary biotechnologist. Please go ahead. Thank you, sir.
Okay, at the outset, I would like to thank organizers, and especially I need not thank Professor Rajendra Prasad. He is the person who is mentor for everybody. So with this, so before I start actual presentation per se, I would like to give only two comments. One comment is that long back in 90s, I used to organize one Mahabaleshwar seminar in modern biotechnology. And in that one, once we invited Professor J.D. Bullock, who was a chief editor of Biotechnology Letters. And we had speakers who were talking only about the recombinant DNA. Then Bullock stood up. He said that only recombinant DNA is not biotechnology. You have to have even dosa fermentation, idli ferment, and he used to publish such type of papers in biotech letters that time. So it is the common biotechnology is important. Our conventional biotechnology is important, which is really appreciated by the end users. Second comment, which I would like to give, recently I organized one radio talk of some farming communities, farmers, and I just asked them, what you want? They said that nobody is thinking about us. And this is the first question I think we should address, that what they want. They really want new technologies, or really they want increase in the yield, or uh, just to reduce down the suicides, etc. in Maharashtra. One has to think about that what they want and how we can help these people. With this, uh, I would like to talk something about the issues and challenges in microbial processes. Now, whatever we are doing is microbial technology. And now I don't want to talk about the bio-E3. I'm just highlighting only 3 points: agriculture, bio-based enzymes, and chemicals, because these are the three areas where we are doing something for end users. Now, about 58% of Indian population has agriculture as a primary source of livelihood. And the change for rural livelihood sector to the modern business model is certainly important, and we should think about that sort of change and how we can go with such type of change. And can startups in agriculture change this scenario? This is the first question. And second thing is that it has been dreamed by our government that we should have 5 trillion economy by 2024. Now it is 25. Maybe in 26 or 27, we will have that one. So how much contribution could be of agriculture community? Now, my first example is of the microbial pesticides. There are different types of organisms which we use uh, uh, as microbial pesticides: fungi, bacteria, and viruses. Bacteria, Bacillus thuringiensis. This is one of the commonest examples. Bacteria and viruses, usually they are effective by ingestion, while the fungal organisms, they are effective by contact. And that is the important thing, that when we spray these infective propagules in the field, we can see that they can kill the insect pest. There another approach which can be as a novelty at a wide spectrum, enzymes which can hydrolyze protective cover of insect pests and the fungal organisms. For example, fungi have cell wall of chitin base, while insects also they have cuticle of chitin base, and we can have the enzymes. That one we are going to talk little later, that these enzymes can certainly used in the combination with other agents to control the pest and pathogens. And third approach is that can we increase the efficiency of these agents with using specific enzyme inhibitors, targeting enzymes which are essential for growth or differentiation or virulence. So these are the three approaches when we talk about microbials. Now, what is a major concern? Actually, this is the process from conidium or the from spore, how it can enter into the insect paste and then how it can kill. But the important or the major concerns are that viability and virulence of mycopesticide. Number of times it is seen that if these fungal organisms, they are saprophytic. They can grow very well saprophytically, but when they fall on the insect paste, they can kill the insect. That time they become virulent, and therefore the balance between the viability and virulence is important, and the yield of the conidia and the virulence is also important. Then effect of temperature and humidity, these are the common problems which industry faces, and the knowledge of the biology of pathogen and host. And here I can certainly say that we are scoring more on this because we have worked on this type of organism, that the insect pathogenic fungi, for almost 20 years. Now, this is the production profile that whenever you want to start any industry, I mean, it is quite simple that you can grow your culture on any grain. We are growing our metarhizium culture on rice, and these cultures, they produce conidia, and conidia are the specific infective propagules. So what is important in this is that the how much yield you are getting and how these conidia are virulent. So I'm just giving both, trying to keep the balance, that if you want to start any industry, it is not the only the volume of the product, it is the importance, the significance of the physiological base of the product is also important. Now, what are the issues and challenges one can face? Is that the first is that, as I said, that you try to identify a strain which is more virulent. At present, the strains, most of the strains, even our strains also kill insect pests within 3 days. Can we identify a strain, virulent strain, which can kill the insect pest within 2 days? It will certainly stop the use of chemical insecticides, because chemicals, they just kill the insect very fast. So we need to have such type of strains. Second thing is that this balance between, as I said, that spores yield. For example, 4 to 5 into 10 raised to 10 spores we get per gram of the spore powder. So if we try to increase that yield to reduce down the cost, certainly virulence goes down. So you have to keep that balance. Third one is, of course, the conidia production cost. In that one, we can say that the microcycle conidation is the new concept. Not this is the old concept, but which we are now applying in while growing these insect pathogenic fungi, where conidium can be germinated directly into conidium, so we can get the more increase in the yield. But again, as I said, that the virulence, that factor one has to consider. Otherwise, to reduce the cost, what we can do, we can target multiple pests with the same preparation, or even pathogens, and thus we can reduce down the cost. Because when we talk about the industry, cost is the important factor. Though we are not talking about the economy, but still, this is one of the main constraints I can say. Protection of IPR. Because here we are using live organisms, so naturally we have to see that how can we protect, because any microbiologist can take the soil sample and isolate your organism and can use. So how we can protect? So we have developed some microsatellite markers, also virulence markers like chitinase, chitin deacetylase, protease, lipase, virulence markers for the protection of IPR. If somebody takes our culture, we can certainly identify our strengths. Strong incentives is, of course, the bioprospecting. In addition to your control in the field, we can have some additional activities. And novel approach, uh, combination of different strains or species or genre, or combination along with the enzyme or enzyme inhibitors. So these are the different issues and challenges we can see in case of using these metarhizium-like organisms. Now, bioprospecting of metarhizium, here just number of examples I have given, that these organisms can be used, their proteases can be used in detergent industry, lipases can be used in dairy and food manufacturing, etc. So these are the different avenues which are available to reduce down the cost. Now, SWOT analysis, because for everything you have to have the SWOT analysis. What are the strengths, weaknesses, opportunities, and threats? Without that one, you cannot really proceed further in the industry. We have different microinsecticide products like metarhizium, Beauveria, verticillium, Pasolomyces, BVM, that is combination of Beauveria, verticillium, metarhizium. So all these, all these products, we have done the SWOT analysis. What is the treatment cost per hectare? What are the weaknesses? That weakness is one of the important aspect as far as the agriculture community is concerned, because farmers do not have that sort of perception for the use of such type of organic product. So farmers education is necessary. That is one of the major weaknesses. Opportunities, it is having growing market, because for example, total chemical insecticide market is around 600 US dollar, 600 billion US dollars. So this much market is available. So the opportunities are tremendous. What is the threat? Know-how cannot be kept confidential, as we are using microorganisms. And main hurdle is of course Central Insecticide Board. We'll see, we'll talk little later about this. Now, the second example is of the enzyme industry. We can see that by 2031, enzyme industry will be flourishing. Around 10.2 billion US dollar industry will be there. Different enzymes are being produced commercially, like proteases, lipases, carbohydrates. In that one, amylase, xylase, etc., polymerases, phytases, and other enzymes like urokinase, streptokinase. So these are different types of enzymes. Agriculture is one industry. Agriculture and wastewater treatment, etc., are the two industries where it is the high volume, low cost industry. While there are certain industries, especially healthcare and pharmaceutical, which is high value, low volume industry. So in this case, we can find that the different microorganisms, hello, we can see that the different microorganisms are being used for the production of these enzymes. I am just highlighting few examples here. For example, protease, it is Aspergillus niger. For lipase, it is Candida lipolytica. These are commercially produced. Chitinases, for example, Serratia marcescens. This is the first product which was used for the control of Pyricularia oryzae. Then amylases, Aspergillus oryzae, Trichotoma rhizae for cellulase, and Bacillus amyloliquefaciens for glucoamylase. So these are the different organisms which are being used in the industry. So what is the uh, thing which in the industry has to follow? So number of times we find that when we talk about any enzyme or something like that, we say that okay, this enzyme is working in the laboratory, but can really it will work in the field? That we don't know, and how we can do the scale up. So all these aspects are important. So one has to have the strain which can produce high titer enzyme. Again, the same thing as I talked about the mycoinsecticide, that we should have strain which is highly virulent. Same thing is true, the strain which has high titer enzyme activity. Such type of strain, if we identify, certainly it can reduce down the cost and industry will take over such type of activities. Then there are different aspects that cell biology, where we can have different cell factories. I can talk little later. And pathway engineering and directed evolution, these are the high level science applications for the industry. Then fermentation scale up. From 10 ml, we can go up to 360,000 liter fermenter. So this is the scale which we expect. Can we have such type of scale ups for our enzymes? Then downstream processing. This is again the cost intensive aspect, that downstream processing, where the recovery of the enzyme is different, is difficult, and concentration, purification, etc. So these are the aspects which one has to think about, and the product packaging techniques, other commercial considerations one has to think about. These are the microbial cell factories, and one example which at present we are using is E. coli for the chitin deacetylase production. Actually, chitin deacetylase is the enzyme which is important in the degradation of chitin of the fungal cell wall or insect cuticle, whatever I say. And then this one we are trying to, it is for the from the fungal organisms, but we are using E. coli for its production. What are the issues and challenges? For some enzymatic reactions, we need cofactors, so we can use whole cells. for such type of reactions. And second is the separation processes usually inhibit the enzyme activity. So in situ separation processes are necessary. So these are the important aspects which one has to think about. And can we use the novel approaches? So for example, efficiency of enzymes can be increased by immobilization. So if you want to work on different enzymes, you can uh, go with such type of challenges. We have one product which is, fortunately for me, DBT appreciated this one as a wide spectrum biocontrol agent to control pest and pathogen in a single crop system, which is nothing but the enzyme preparation. This enzyme preparation is the combination of the mixture of different enzymes like chitinolytic enzymes, chitinase, endochitinase, N-acetylglucose. This is the complete setup, I can say. Lipases and proteases, because lipid protein are the important covering agents in insect-cuticle. Same thing is true for a fungal organism. So these enzymes, and in addition to that one, glucanases and mannanases. So one organism produces all, this is the cocktail of enzymes. So we are trying to work with this type of enzyme system. And we had checked enzymes in the field also for the control of different pests, woolly aphids, mealybug, Helicoverpa armigera in pulses, Spodoptera, then fungal organisms like Fusarium, Sclerotium, then downy mildew, powdery mildew. These are the obligate pathogens in grapes. So even we had tested against the nematode, etc. So now the application is there, but the cost of product is very high. So that one we had to now reduce down the cost. So different approaches we are trying to see that can we have the nano formulation which can reduce down the cost. So that is one thing at present we are working on. Then the transportation of large volume is not feasible. So we have to have some sort of concentration mechanism, and shelf life, increase in the shelf life is also important, and the stability. So again, when we go with SWOT analysis, you have to see that uh, it is compatible with other integrated pest management components, and it is biodegradable. This is the proteinous material, so it is biodegradable, so it is not harming the field per se or any other organisms. Unfortunately, what is the threat? It is always compared with chemicals. Even when we wanted to have the toxicological study, it was compared as it was considered as chemical, and all the toxicological data generation for chemicals we did for the enzyme, and fortunately we got the clearance for that. Third aspect is about the chemicals. What we can say that it is antifungal drug market. And here we see the main players are azoles, echinocandins, polyenes, and Anand must be very happy when I say that about the chitin synthase inhibitors. Mm. So it is chitin metabolism inhibitors, because why chitin? It is the main structural component of insect pest and fungal pathogen. So there are two different aspects. One is the synthesis of chitin, and another is the hydrolysis of chitin. We need hydrolysis because for the further growth of the any fungal organisms, we need a hydrolysis, and the hydrolyzed products are being used for the further growth of the organism. So chitin synthase is the key enzyme. Another enzyme is glucosamine 6-phosphate synthase, and two enzymes for hydrolysis, chitinase and N-acetyl glucosaminidase. Now, these antibiotics, antifungal antibiotics, are produced by again actinomycetes. And so at present, we are working on the different inhibitors, which we can use in addition to our metarhizium and enzyme preparation. So in agriculture, nicomycin has been again used earlier independently for the control of Pyracularia oryzae, that pathogen. And now we are trying to use this nicomycin to reduce down the cost of our preparation in combination. And why chitin synthesis? As I said, that it is present in all fungal organism. It is not present in humans and plants, so it will not be toxic to human beings. Now, with the help of the organic chemistry division, our NCL group, we are trying to modify the nicomycin molecule at different positions. And we have now novel idea that instead of monomeric inhibitor. We can have dimeric inhibitor. That means the double the action will be double. So we are also working on this aspect. And the third one is a novel approach is that chitin synthase inhibitor and glucosamine 6-phosphate inhibitor. Both are joined with some spacer, and we are trying to have such type of molecules to control the different pathogens per se. Two other enzymes, again, we are doing some work, glutamate dehydrogenase and ornithine decarboxylase. And we find that the one NADP-GDH inhibitor for our organic chemistry division, they have identified one molecule which can certainly show the inhibition of different fungal organisms, especially Candida, Candida glabrata, our Benjaminella poitras. But we have not tested this type of inhibitors in the field because it is, as I said, in agriculture, it is a large scale, large volume is necessary. So we have to produce this amount and large amount of this type of inhibitors, and we are requesting our organic chemistry division for that. And ODC inhibitors at DFMO, this is being tested in the field for the Puccinia, Verticillium, Rhizoctonia, and Helminthosporium. So these are the pathogens per se. Now, before one starts, every concept or experiment cannot be commercially feasible in the given time frame. It doesn't mean that it is not feasible. According to me, it will be feasible, but the time frame should be different. Then process scale up. One cannot simply enlarge laboratory scale. I think this is for the young entrepreneurs, those who are interested in going for entrepreneurship, that duplicate lab scale conditions at large scale. It is not just only the lab scale and duplicate that one. Then important point is that begin with the end in mind. Be diligent in the details. That is based on the realistic biology, chemistry, and engineering assumptions. Build process flow diagrams, material and energy balances, unit operation designs, and technological models. So all these things are necessary if you think about going for the entrepreneurship and prepare for unexpected. For example, we started in November 2019 and February 2020 because of COVID, everything stopped. 2 years we could not do anything. Of course, I did something that will be the last slide. Now, we are now trying to develop one platform for translational research in agriculture biotechnology. It is not possible for everybody to have his or her own industry. I did because uh, I don't know, but somehow I'm quite passionate about this. Uh, what is that platform is going to do? That platform will help people. If you have good proof of concept, you come to us, we can go for the scale up and further up to market, we can take you up. It is like venture studio concept. That means we will work with you together, and then all the finally successful project is important, and we can help the any entrepreneur for such type of activity. At present, we are mentoring AU, Assam Agriculture University, for such type of startup. We are working with them for the insect pathogenic fungi. They are developing different products, and we are evaluating them in the field, and we are trying to help them. Consultation to the farmers, free of cost. That is important aspect when we start industry. And popularization of your concept is also important. And that's why I usually give radio talks in Marathi, of course, for the farming community. And thus we popularize our products. So in 2 years, what I did during COVID, we could publish the Springer Nature publications, 4 books in this 2 years time. My recent book is Fungal Biopolymers and Biocom. Of course, these are the edited volumes. I have a couple of chapters in this one. So this is the at least thing which was unexpected COVID, but we could come out with some type of advancement in the mycology and mycotechnology knowledge. Thank you so much for your kind listening.
Thank you, sir. This is a real translational research,
Yes.
and I'm very happy. I hope you had some more government support really to make it
Yeah,
big.
then we can increase the facility. Otherwise,
But
whatever production we are doing, we are using that money for further development.
any quick question?
Thank you. You really showed that we need important biotechnology more than fancy biotechnology. But a specific question is that the lyophilized enzyme that you talked about, what is the shelf life and how do you plan to bring it to field modern, maybe how do you think you can transport, you can sell?
Yeah. Actually, that's why it is not lyophilized. We can say that we can have the any other mechanism. For example, ammonium sulfate precipitation. If we can have that volume if from 10 liters, if you can come down to 1 liter or something like that, still transportation will be easy. So we are trying that one. Then again, another lyophilization we tried, but lyophilization is a very costly affair. So we cannot really use lyophilization. So by absorbing these enzymes on different polymers. For example, chitin chitosan. We are using that approach also for making the powder along with this. And chitin chitosan, there it has a lot of advantage. Is that when we put that one in the soil, it can increase the chitinase production potential of different microorganisms. So we are using that. And spray drying is another thing, but still it is not that much convenient.
Okay, sir. So
Can-
nice. I liked your talk. So that cocktail of enzymes that you showed, so make it to in one together.
No,
It's a
it is
different-
only one organism. It produces different enzymes. Actually, I can tell
Mm.
the name. It is Myrothecium verrucaria. And fortunately for me, DBT supported that project 3 times. And they wanted me to, even toxicological studies for that one also DBT supported. They wanted me to commercialize, but unfortunately, because of the cost at present, we are working on that one. Maybe by next time when I come, that time it will be commercialized.
Thank you, sir.
Dr. Deshpande, I'm back here. If you look behind, I put my hand up. Yeah, it was a very interesting talk. I was wondering this idea of, farmers interfacing to bring out questions that they really want, solutions for. Are there, programs that actually do this in the country? Are you aware of any? And because you thought about this, I was wondering
yeah.
whether you.
Actually, I mean to tell you most frankly, it is RGSTC, that is Rajiv Gandhi Foundation, our state government organization. They are interested in social significance of any project. So earlier we submitted one program to RGSTC and we got the support. That eventually I found that it is not sufficient. We have to bring farmers in always. And then now at present, we have all the social media. We have 10,000 farmers on our on our WhatsApp group, different WhatsApp groups we are combining. So in that one, we are launching our different ideas. And so recently, Pune radio station, they asked me that whatever work you are doing, for example, just 1 second, sir, it is nano urea, which is not at all good concept as far as the requirement of the crop is concerned. But somehow it is given, and now it is made compulsory that 500 ml of nano urea one has to buy. But anyway, so that time farmers said that what we want is important. What you are developing is not important. So that's why then I had that radio discussion forum, and we discussed all these points. But I don't know how much effect it will have. But still, in Maharashtra, at least it is there.
Okay,
Good afternoon, sir.
so I would like to thank you.
You had to come, or shall I come?
No, I come.
I'm taking this much liberty with. बंद करा तो
Yeah. So next talk is online. That's why it's taking. Oh. Oh. Good afternoon, Sundar Singh.
Good afternoon, sir.
So he's our next speaker. Most of you must be knowing him. He is our Genova Pharmaceutical CEO in Pune. And in short, he is known as Vaccine Man, mRNA Vaccine Man. He has set up a very good pharmaceutical concern. And Sanjay is slightly rebel type. So he spent a lot of time in NIH and left the job, a good job, come back to Genova. And since then, he has been taking Genova to great heights in terms of mRNA vaccine at the COVID pandemic time, and also biosimilars. And he has many, many products which are in the societal issues. Sanjay, it is a great pleasure really to see you. I don't know, where are you now? He was
Sir, present in Pune itself.
okay. He was supposed to be in Tokyo today, but something happened. And Sanjay has been a great support for us in MIT. We also have a MOU in place. In fact, there is a very competitive internship offered by Genova, and many of our MSc students compete for it and have been going for 4 or 5 years. So thank you for supporting this conference also, which I must acknowledge. Thank you is not enough. So I give you podium to speak on your recent. Yeah.
Thank you, sir. Thank you, sir.
Go ahead.
So sir, I have to load my presentation, and is the way to share the screen? And I've also sent my presentation to you on the email. If that can be loaded, that will be very helpful, and I can only ask the next slide. But in the meantime, thank you very much for inviting me for this talk. And by heart, I'm still a scientist, trying to do the work. And in busy life, like trying to write few papers, because science need to be supported by good publications, reviewed publications, and also the product need to go to the market. So in this whole process, I think the opening session was very good by Professor Bamjai Saab, and he raised certain very, very sharp and important questions. And I made a list of that. I'm just going to touch one by one. So one is that in organized ways, there is academic and industry collaboration. Is the biotechnology is limited to cellular and molecular biology? Is the CCMB kind of academician? Is the tangible outcome is from our biotechnology? Is there is something local which innovation can go to global? Is we have to come out of the comfort zone. He gave example of one of the disease which was solved where 11 teams were involved. He also talked about the Cas9. Is the bioeconomy is killing the innovation? Catchy slogan, platform technology, patent converting into product, diagnosis and solution, and something about few lakhs people in lakhs of village, and how we can have a ground level impact and some biology and evidence-based medicine. So I think he has made a very good point. And sir, is my presentation is available? Is I'm audible, sir? Hello. Is I'm audible?
Yeah, you are audible. Only thing is your presentation is not downloaded here yet.
Is it getting downloaded? You have seen the mail or not?
Yes, I've seen.
Okay, so if that can be downloaded, so in between I can talk for a few more minutes because looks like there's some limitation. I cannot upload my slides. Maybe from my side, it's sensitive system. So, but it is very important. I'm a trained biochemist, and I'm many years, many maybe decades junior to Professor Prasad. My hairs are much more grayer, but I have from day one, I was very passionate about my interest in applied biology. So did my master's, did PhD at Central Drugs Institute, worked a couple of years at ICGEB, went to NIH, tenured, did my work, but everything was looking to me was not fitting to my schedule. So it is better to start your own. So this is where we started Genova. Our primary focus is from day one that whether our scientific innovation are making the societal impact, and where I want to bring this young generation thought process, how we are doing, what we are interested, how we should modify or update our teaching, how we should modify and update our research, and how academic and industry collaboration should work around, and where we can end up as an organization or as a company or academic institute, which make a different discipline and which makes a deliverable clarified for society as a taxpayer's money or it's what you call shareholders' money. All ask these kind of questions. So I think I'm very keen. I want to take another very simple example where we do. Even as a profit organization, we work on leishmaniasis. And today I'm focusing my presentation on leishmaniasis. And you might see that I'm trying to answer all the questions which Professor Bamjai has asked. He's a Padma Awardee, very respected person. But sir, we are trying to answer most of the questions, though the presentation was made before, but looks like we are trying to walk on this path. Professor Prasad, is the presentation got loaded? I cannot hear back.
Well, it is getting downloaded now.
Okay.
Pardon me.
So with these words, I'm looking forward to have and my presentation is not very long, and I've tried to make for students very quite simple. And then I would like to have some open questions if students are keen to ask, and I will be very happy to answer them. I think the presentation is up. I can see it. Can we start, sir?
Yes, sir, you can proceed.
Okay, thank you. So my talk is based on the critical role of need-based biotechnology R&D. It is very, very important for us, and one of the example we are presenting in front of you, and we believe that that will be if we focus on this particular topic, starting from very early days when we are a student, we can look for shaping the future of the country and how we can look at the Indian perspective. Next slide. So here we are talking about leishmaniasis. If you can see, this is a disease which is spread close to 88 countries. Around 1.2 billion people are risk. Roughly, we get close to between 5 to 1 million cases new years. 12, 15 million people are infected. 50,000 deaths as recorded by WHO, and it expands its footprint. And currently, there is no vaccine. And a couple of times, many countries have said they have eliminated leishmaniasis. But if you see the picture, which has one of the publication which we found, looks like it is most of the places, and this is the disease is in three forms: visceral, cutaneous, and mucocutaneous. In India, also this disease is endemic. Certain regions in the Bihar, it is there. In Odisha, it is there. In certain part of the other, in that tribal area, it is there. But basically, this is a global problem, and we thought that can we be part of this eradication process, because there is no currently available vaccine against it. Next one. Next one. So there could be two possibilities. One is, can we detect the disease where it is spreading, because there is a medicine available for it, liposomal amphotericin B, which you give it, the disease is spread is limited. And one of the big center we work very closely in BHU, which is the reference center. So disease detection is very important. And if we see that in three particular villages, there are people, can we detect who is infected, who is not infected, who is immune? So we need a detection kit, which is very, very limited today. But simple detection, just like tuberculosis, PPT. Next one. And then it is not big spread. This is quite endemic. So if we have a vaccine possible, we can eradicate it. So this is what we are trying to do. We have a live attenuated leishmania vaccine program at UNOA in collaboration with many local and global partners. Next one. So disease detection, we have developed a leishmanian skin test. This test was available, but in very undefined format, and maybe 15 years back, it gone away, and it is availability is very limited. And this program we started on the behest of WHO primarily. Next one. So if we want to decide why we need LST, the first one is LST is better to exposure and immunity to the leishmania parasite. LST can be used as a tool for surveillance, epidemiology studies. Next one. And unavailability of this is GMP grade, which is more defined grade, is not available in the market. So this is we need. What we need exactly a well-defined strain of antigen production system. So it is controlled, defined as per modern CGMP take. We should have a defined cell bank from which antigen has been produced. It should be robust, scalable, and then it should be in such a format which can be made available and can be used globally, especially the places where it is less like Sudan or Ethiopia or Kenya or Nigeria or other part of Iran, many places in the world where it is used, that should have stability, because antigen which was before available and it wiped away, it was need to keep at minus 80 degrees centigrade, which is not feasible, and modern science can support this. Next one. So we started this collaborative work. with one of our collaborator who is based at USFDA. Whole life he has worked with on leishmaniasis. His name is Dr. Hira Kasi. And they said that they were having some process, but that process we grown the parasite, and this is very well defined parasite at 1 liter, 5 liter, and 10 liter scale. And we said that there is a continuous process can be developed, which is well defined as per CGMP norm, and stability, growth, all characteristics we have made it. Next one. And then we develop it by, as I said, if you see on the A column, it's the bioreactor system. We made it, harvested, washed, cleaned it, freeze thaw, and made a formulation and made it -80, the way it was available before. If you see, if you see the D part, it is a very, very busy lane. But now with modern tools, you can characterize everything. And then when we made this one, we immunized the hamster, and there are certain criteria which we measured on the right side. If you see, we found that it is creating the detection possibility, and it is showing all the clinical symptoms which is required. And this paper we published in Nature Communication, and because of the students should not get confused in too much of data, so most of the data is in the paper, and you can go and you can have a look into it. Next one. And once we develop the proof of principle, then we realize -80 is not feasible. So we develop the lyophilization form, and it is a couple of years of effort. And finally, the screening has been done, and one of the formulation is getting tested in Brazil, Sudan, Sri Lanka, in Bangladesh, and many more countries through the help of WHO. So let's see, initial POC can be tested, and then we can go for next one, which is make the CGMP grade. So here, one of the major, most important data, which is, you will see the manuscript will soon come into publication, that if you test as a PPD kind of model, this on the left side was liquid formulation, on the right side is the lyophilized formulation, and there is no difference, but it looks absolutely fine, and it is giving the right set of data. So at present, we are testing it in the field, not immunizing the human, but taking the PBMC and how response will come. Next one. So in a very simple way, I have presented. So we have developed the lyophilized formulation, POC established, GMP manufacturing is ongoing, and we are in discussion with regulatory agencies like ICMR, CITESCO, and WHO for this. And we feel that this will be one of the very important tools for leishmania eradication. And I have shown you one of the WHO slides down the line. Next part. So if we have to characterize, so this is the one of the format, you can see so many things we have to do on the cell bank. So it was very difficult for us to make the CGMP cell bank. This was done back at ATCC in US, and they defined the parasite, they characterized the parasite, and finally parasite was handed over back to us, and this is what we are going and making it. Next one. Now, yes, you can detect the disease, you can see the epidemiology, who need to be vaccinated, who need not to be vaccinated, where the disease is there. This all can be done. That was the diagnosis, as Professor Bamjai said, that what are the tools for the diagnosis. But now, next one, can we vaccinate the people who are at risk of getting infection? And for this, we worked this is like 5 years before work has been published. This was published again in Nature Communication. Here we use the CRISPR-Cas9 technology to knock out a particular gene, and then we characterize the whole genome that everything is fine. So this knockout of particular central gene, what happens that in vitro, the parasite, if you grow with time, it dies, like if you see on the in vitro growth curve. And in the second one, in vivo also, if you give with time, it goes into macrophage but die by its own. So it is not creating the disease. So we characterize this, and this POC we published again in Nature Communication a couple of years back. Next one. And then from there, how to manufacture this live attenuated parasite, which was very difficult for us. So the Gates Foundation and some other agencies, which is GHIT, we reached to them, and they said we are ready to fund the program. And let's see, we have the expertise of mammalian culture, so let's see whether we can do it or not. Next one. So we developed it and made the parasite and GMP level and GLP level and grown the parasite 1 liter, 5 liter, 10 liter. And finally, we succeeded that this is creating the same way of what was doing the lab grade. If you see the parasite burden in a immune compromised and non-compromised, you can see this is less than centrin where we have immunized, there is no parasite growth. And also when the scent fly comes back, it cannot pick up. So the parasite just goes away. And again, further next year, we publish this paper in Communication Biology, which is also Nature Springer Group. Next one. And then we also tested with cross protection whether it is also going to protect the cutaneous other than visceral, and we found that it is effective very well in the animal model system. Next one. And then finally, GLP toxicology we have done where we can see the vaccine is giving interferon gamma response, IL-10 response, interferon gamma IL-10 ratio looks good. Next one. And then in toxicology model, we also use the second thing with the rat, and where we also measure the functional immune response in form of CD4, which is interferon T cells, CD4 TNF, CD4 IL-2, CD4 IL-10. And all this was years of work with us when we looks like that this vaccine is giving protection based on the CD4 T cell responses in rats. Though we have not challenged the rats because rat is not a model, but hamster is the model where we have already published it before. Next one. So now we moved into GMP manufacturing 2 years back. We go ahead, next one. And again, this knockout parasite need to be characterized. It's a huge program for us. It takes around a year to characterize the parasite, make the CGMP grade parasite, and it is available for 100 years. So this is the qualification of this. This is the industrial complication which I wanted to present to the students. Next one. And this was a very simple process. We take this parasite from the cell bank, which is well characterized. We grow it. Finally, we take to bioreactor. From bioreactor, we can do the harvesting. We can clean it, everything, and we can make the vaccine formulation. This was the GMP process, but it is available at 1 liter, 10 liter, and finally produced at 50 liter scale. So few million doses is available. Next one. Next one. So this is when we went to WHO and discussed that what kind of vaccine target product we required. So it should be cross-protective. It should be go to all endemic regions. Efficacy should be more than 70%. Preferred is 95%. Safety profile should be there. Number of doses could be 2 or 1. IMID route, 1 year or lifetime protection. Product stability, they require 12 months, and vaccination single dose vial. And they also, the new thing came out, and which is very, very important, that we focus ourselves that can we make it affordable. So the preferred, our price, it should be less than $5 as compared, why I'm saying dollars, because it is a global currency, so we may quote it all across the world. Next one. So that was the required if we qualify the vaccine, but these are the required, these are the vaccine specification. We found how it appears, what is the parasite level available, what is the viability available, how they are responding to different assays. All these things were processes were developed, assays were developed, validated, and then moved forward. Next one. So if we want to say in conclusion, we established the immunogenicity of this gene knockout parasite. We removed the antibiotic marker. We developed the vaccine, go into the bioreactor, develop the stability studies, the immunogenicity, cell banking, GMP manufacturing. And we are very happy to tell you today that on 27th or 28th of August, this is made in India product, we got IND approved by US FDA, and now the Brazil and Kenya is also approving. So this is basically local level work at global level competence, and hopefully it will make a difference over there. Next slide. We are we should also publish well, and those are also important. So our work was first featured in Nature in 2023, and then keep on going. So this is Nature Vaccine, the work which I have presented, have been published there. Next in the dog model, next one. Then this is what we wrote about the technology. Next one. This is communication biology, where preclinical validation has been done. Go to the next one. This is the nature communication, where LST skin test has been, we had said that we can reintroduce in the field. Next one. And this is manufacturing preclinical toxicity of GLP grade this. So basically, we just wanted to, this is for young students. Any work you do, you have to maintain a class and you have to maintain a global competitiveness. It is very, very important. Otherwise, we will be not counted. You have to ensure that you get counted, and the get counted is also the, and we have filed around 7 patents on it during this process. Go to the next one. And this was not the work which was been done, Genova. This was the whole big collaboration. It is US FDA is involved. You might have been hearing a lot of news about US FDA, but you have never heard that US FDA is working with a company back here in India. McGill University was responsible for the knockout. Ohio State University was responsible for some assays. At present, we are developing the human challenge model at NIH Clinical Center. Most of you must be aware of it. NIH Clinical Center, we have the center, has a center in Clinical Center where developing the human challenge model. John Hopkins, Cincinnati Children's Hospital, and our Brazilian partners there, Pasteur Institute of Tunisia, which we did our top trials, and Nagasaki University, where we did most of our animal experiment. And this program has been heavily funded by GHIT. Few dollars we got from Wellcome Trust and NIH, but this is Genoa internal funding and GHIT funding. And I think my last slide is thank you slide. Next one. So thank you very much. And on the right side, you see that we have also tried to qualify that we are one of the best places to work. And this is end of my presentation. Now I'm open for questions. Thank you.
Thank you, Sanjay. Very informative. And we are looking forward to your products, of course. Some questions you would like to answer?
Yes, I will be very happy, sir.
Wonderful talk. I'm Syed Hasnain from IIT Delhi. We met the other day. I just wanted to check with you. Are you working at some other disease also? Leishmania is certainly a big issue, but are you looking at some poor man's disease such as tuberculosis, or it's not on an agenda?
So last week, the paper had been accepted in Cell. Hopefully, it should cover the cover page with my collaborators. It's a joint collaboration between Harvard and Genova. And very soon we are going to enter into the phase 1 clinical trial. So we are working on the TB vaccine, and it is on the mRNA platform. Another 2 more vaccines are there, and this is rabies, because you might have seen rabies vaccine needs some modern look. So that will also enter soon into clinical trial. That will the fastest maybe within 3 months. And then another a global program funded by CP, and we are working on the Nipah vaccine, because every time when Nipah hits, the whole country becomes standstill. So we are also working on the Nipah vaccine. These are the certain vaccines which is of our importance, local importance, but also global need. We are working on it.
Well, Sanjay, congratulations. I'm Bamzai.
Sir.
2 observations. One, infectious diseases in general in the country, we have been doing well. Vaccination, and you have done extremely well, both from publication point of view, which is basic science, the whole process systematically. I think it augurs well with the students here. They know how they should carry out their work. So thanks for presenting it in that fashion. Second, taking it to a logical end where you have a vaccine against Leishmania. But the question now is, vaccine programs, we have been more or less doing well in the country. But when I mentioned about other sets of diseases where intervention has not been possible, and that is genetic diseases, rare diseases. So I think if now that you have this handle and the access and the way you are going, I think if you attempt in due course of time, you have your own priorities, but in due course of time, if that happens, we'll have something local along with global. But the other observation is the collaborative activity. Obviously, science is collaboration. When will we be self-sufficient to carry out all the features which you talked about, where you had to collaborate with others abroad, that we do it within our country? I mean, that's another concern which I have.
So this was one of the program where I said I have these my collaborations, but the way we keep on doing, because this is limited 30 minutes. I wanted to give a best crisp for students, but we work with very closely with Professor Vijay Chaudhary, who will be, I think, today or tomorrow. His talk is there. We work closely with him, and we develop one of the largest fast display library back in Genoa, and a CFV library, human fast display library, human SCFV library. So we collaborated it. On the rare disease, looks like you will see some wonderful news soon. We are collaborating internally on hemophilia A program, and then yes, we have a vast portfolio, but we choose our priorities very carefully, and we want to see the end. So when the day we start, we want to see that whether we can we can treat 10,000 kids with hemophilia A. So that's the program we started, and it is again a high-end cell therapy. So we have these programs internally also, but today somehow this program was what I presented as example, because it is very important for us somewhere in our mind that we should solve the problems. Biology was not in the best scenario till pre-COVID. Bureaucracy, IS, politics, everything. But when COVID came, everybody was inside the cell, then only the biology came and solved. So we came out. So I think we have to, every time we have to go and prove it ourselves. It is our responsibility as academician, as a scientist, as industrialist, that we make the difference in the society. And that will take a bigger impact. So whenever I get a chance to go to university, I take my time and go and give a talk, because I want the scientific temperament is very important, but scientific temperament should be in the way that we have to solve our local problems first.
Hello, good afternoon, sir. So you talked about visceral cutaneous and mucocutaneous leishmaniasis. So does the vaccine target all the three differently, or
All the three, it looks like it is working against all three equally.
so the mechanism of action is equal in all three?
Yeah, yes.
Okay, thank you, sir.
So sorry we were late to start with, but thank you for your time for this wonderful presentation. And we are very hopeful that you will give us many more products for human health and disease to prevent, of course.
So thank you very much for sir organizing this meeting. It is very important, and we are so proud that from JNU you went to the private university and you have done so well, and so many publications, so much science, and so much students. Thank you very much, sir.
Thank you, Samir. Thank you. So we break now. We are late, so only 15 minutes. And those who drink tea, come back. Well,